So, hello everyone. Am I audible and my slides are visible? Am I audible and my slides are visible? Please let me know. Should I start? Please give me a confirmation that I am audible and my slides are visible. Okay. So, last two topics are left of the last module. So we have covered this one, the NGS and the bioinformatics role in microbiome research. Now, today I will be focusing on this part, AI and machine learning in microbiome based diagnostic. And I will also try to cover this ethical and regulatory aspect. So if not possible, we will do this tomorrow. So now what is the role of AI and ML in microbiome based drug discovery? So if you talk about the artificial intelligence or machine learning, then you must be knowing that life has been easy and hassle free. Everything is possible just on a single click. You don't have to put a lot of effort. You don't need a lot of labor. You can do just by having some of the easily, or you can say freely accessible softwares or web server. Everything is possible. So what is the role of this AI and ML in our context? Our context is what? Microbiome based drug discovery or microbiome based therapy. So AI/ML is used for the microbiome data mining through the bioinformatics. So this is nothing but it will be correlated with the second topic, that is the role of bioinformatics in gut microbiome research. So we are having huge amount of data generated after sequencing. So that data we need to analyze it. We need to find that whether it has similarity or not. So for that we always go for the data mining through the blast and through different softwares. So there are so many different softwares available which will let you know that if you are putting any query sequence, the sequence which we put is known as query sequence. So if you want to see whether this query sequence is already existing in the database or not, so for that you will run the blast. You will put your query sequence as an input, then you will run the blast, and after that if you got a hit means this sequence is already similar sequence is existing in the database. So that is known as a bioinformatic term, hit. So you will got so many different kind of hit as per the similarity percentage. So higher the similarity will be from top to bottom, like 99.12, 99.01, 98, 97, and likewise. So for this purpose we use artificial intelligence and machine learning programs to make our decision related to what pathogen is this, how can we target its genome, because ultimately we have to target the pathogen genome to kill it. So for that we use microbiome data mining softwares. Now the next part is microbial biomarker discovery. So the microbial biomarker discovery is what? As we already know that if for a healthy individual we are having the composition like A, B, C and D, but if you are an individual who are who is not having the natural gut microbiome composition, or you can say the diseased one, so it may have some other microorganism, let's say E, F, G, H, along with this ABCD. So these are acting as a biomarker because they are indicating that this person is not at all healthy. He or she may be suffering from type 2 diabetes mellitus, you can say, or you can say any kind of liver disease, fatty liver, or all those things. So many a times these increased or the you can say varied composition of the microbial diversity indicates that the person is suffering from a metabolic disease. So for that also you will be looking into some database, like if a patient is suffering from this type 2 diabetes mellitus, then what are the chances of its alteration in the microbiome profile. So for that, if you write it down that there is an increase in the composition of aerobic bacteria. So in the software you will be feeding that for type 2 diabetes patient, the composition of or the population of aerobic bacteria will increase. So this way also we are providing database so that it will be helpful for each and every individual associated with microbiome research. So we in research we help others also. So we provide our data on online mode, like all the bioinformatics data is need to be uploaded before the publication wala part. If you want to public or publish any paper, any research paper, so you need to publish all your sequencing or all your bioinformatics related data into already available software, that is NCBI. NCBI is a National Center for Biotechnological Information, of the biggest you can say platform at which you have to deposit your each and every sequence. So this is an authentic website where you need to deposit your each and everything related to bioinformatics. So for that also we use AI ML. Now predictive modeling of drug response. So this is through the protein or gene modeling, docking. Suppose the gene structure is like A, U, G, C, T. Suppose this is the gene sequence, nucleotides. So if you want to know that whether this will be, if you are taking any particular drug and this drug is capable of killing or anyway altering this sequence or not. So you can predict through the docking study that your drug is capable of killing the particular genome of the pathogen or not. So for all these purpose we need the different softwares which are freely available. Many of the softwares are freely available and some are subscription based. You have to take the subscription in order to have some uh, major usage. So all these things require the AI and ML. Now for second part is designing live biotherapeutics. Live biotherapeutics means probiotics. So if you want to design probiotics, then also you need some engineered bacteria. Like if a bacteria is not so healthy, so you need to engineer its strain and to remove all the toxic gene it has in its genome. So for that also you need to engineer it. And how you can engineer the bacteria? You need its whole genome sequence, and that is also harvested from database. So everything, whenever you do a good research means an advanced research. So you will take help of the bioinformatics. Bioinformatics is nothing but the freely available all the database related to protein, related to gene and SNP, mutated one. and everything is freely available on NCBI and other platform also. So you can see, you can say that for designing any kind of probiotic, for any therapeutics also, personalized therapeutics means it will be varied from one person to second person as per their genome or as per their microbial diversity available in the gut. So you can design personalized therapeutics. And this third point and this sixth point are uh, can be correlated. Drug microbiome interaction prediction, this is nothing but again docking study, that whether this drug will be this, this is the active site and you have this type of drug, so whether they will be interacting or not. So all these things are predicted by different softwares we have available. So its AI role is to stimulate microbial metabolic pathway and drug transformation. So for all these purpose, we use the AI ML. Now for personalized therapeutics also FMT, and FMT is done as per the patient specific data. See, specific word will be always used because as I said, the personalized therapeutics means the therapeutic which are provided to person one may or may not be similar with the patient too, because we are designing the therapeutics as per the individual person's requirement and not generalized medicine we are providing to each and everyone by seeing the symptoms. It all depends on the genome we are having. So these are the things in which we use. Now uh, these are the steps like the sequencing, we go for the sequencing, and the microbiome data when we go for the billions of bacteria at a time, that is known as the metagenomics. Metagenomics means the genome of each and every bacteria of our gut microbiome, as we are studying in the context of gut microbiology. So what is the role of AI ML in the data pre-processing taxonomy? So you can see, as I explained, that for the data processing we do use the AI ML because we need some database that can use to analyze whatever sequencing data we have in our hand, because without any analysis that is of no use. Raw data is of no use. You have to process that, you have to derive meaningful interpretation out of that. So for microbial profiling, that you want to know that whether this bacteria is from which phylum, so for that you need to go for the sequencing, and for that you need the role of this one. Now if you want to check whether the group of, let's say group A bacteria is abundant or not, whether there is diversity variation or not, so we are having some classifiers, we are having some categorization based software that will let you know that these are the compositional analysis, these are the uh, composition of each and every bacteria, like the bacteria A is of 30%, bacteria B is 20%. So all those things can be done by the machine learning programs or softwares we are having, and accordingly we can say biomarker detection. Like you can say for the diabetes mellitus patient we are having 30% of A bacteria, 40% of B bacteria. So like this way you can say. And if you are talking about the drug prediction, like if you want to do the personalized therapeutics, therapeutic design, so for that also you can rely on the AIML programs. Now for monitoring means if you are giving any kind of treatment, any personalized therapeutics to any of the patients, so you need to track record all of his or her data. So there are some softwares which are time based, time foundations are there, that it will be tracked, let's say for 30 days, all the parameters will be tracked up to 30 days. So also it will let you know that whether the disease is disappearing or whether it is its progression is going on. So through all these things we you can say by the help of this AI ML the research is up to the next level. Without any help of bioinformatics, or you can say computational biology, everything will become very very difficult. So all those things are very important, you need to write it down in your notebook. So you can see machine learning can help to have personalized therapeutics, so that you can say the therapeutic efficacy can be increased, because the more the efficacy of the therapeutics we have developed, the more are the chances that the disease will be cured. So personalized recommendation as per the patient's requirement, patient genomic requirement, patient metabolomic profile. Metabolomic profile means what the patient gut microorganisms are producing. So you can see here it is metagenomics, metabolomics, clinic. And if you are referring all the profiles of a patient, then accordingly you can, with the help of this machine learning, you can design the therapeutics, you can suggest some personalized recommendation. For example, if you can say that you need to have a probiotic of this kind, you need to have prebiotic of this kind. And apart from that, this is very important that you can let others know that if you are having any kind of metabolic disease, then you will be having different composition of the gut microbiome bacteria. uh, if you will compare the gut microbiome composition of a diseased one and a healthy one, there will be very variation, you can say a lot of variation you can see. And patient stratification. Patient stratification means you can categorize the patients on the basis of what, how can I explain this? You can categorize the patient on the basis of what they are having in their gut microbiome. Suppose this blue one are having the altered bacteria up to 30%, let's say. And if you can see this brown one, you can say 80%. Or on the level of dysbiosis, you can categorize, you can stratify them. Like these are the group of patients who are having the dysbiosis level 1, dysbiosis level 2, dysbiosis level 3, on the basis of the diagnostic report they are getting. So if you will classify them, you can easily say the doctor will easily treat them. It will be very much you can say hassle free. They will simply call that group 2, group 1, or the unit 1, unit 2, unit 3. So likewise it will be very easy. Now phenotypic prediction. So as per the gene, you can gene, this is known as a concept known as the gene to phenotype, because whatever we are just because of our genes. So gene directly provides the phenotypic. It means uh, if I will be having a disease related gene, then the gene will be expressed, because gene always express. So a gene will be expressed and I will be having that particular disease, whatever be the disease related gene in my genome. So if that gene will express, I will be having that disease. So this is known as gene to phenotype. So through looking at the whole genome sequence data, WGS data, you can predict that this is the whole genome sequence. This is the genome, and if you go for sequencing, you will get this stretch. So you can predict that this gene will cause this disease. This gene is for physiological mechanism. This gene is for controlling the disease. This gene is for fighting against the pathogen. So likewise, so you can predict the function of each and every gene after the sequencing by looking at the data we are already having in our database. We are having different kind of data, like all the gene related data, all the function of the each and every gene, all the things are available on a single click. So these are all the benefits of, you can say, advantage of, or the applications of, it will be more appropriate to call it as an application of artificial intelligence and machine learning to help people to get cured by uh, taking the help of all the available web-based server, you can say, or software, you can say. Now this is a little bit more detail. So if you will compile the medical history of a patient, the genetic data means sequencing data, and lifestyle information, like this patient has a sedentary lifestyle, this patient has an active lifestyle, or this patient usually take carbohydrate more, this patient always take a non-vegetarian diet. So all those track records, if the doctor will always ask you all those things whenever you visit a doctor. So everything will be tracked for each and every patient. Everything will be written down. Every little information is required by the doctors. So when you provide all those required information, and then you provide the gut microbiome signature data. Signature data means what? Whenever there is a word like signature, signature means the specific one of yours only. Like I will be having some other microbial diversity, you will be having some other microbial diversity. So signature data means only of that particular patient. So if you will provide this information also and this information also, so what AIML can do, they will do the data processing part, and they will detect the patterns in the gut microbiota and patient data linked to, for example, atherosclerosis risk. So this is an example of only a single disease. You can use all these data, like two types of things are there. First one is your, if you are providing your doctor the gut microbiome report, like your gut is having this percent of this and that bacteria, you are providing to your doctor. Now your doctor will ask you whether you are taking antibiotic or not for a long time, whether you do exercise or not, whether you are obese. He will check your weight, he will check your all, he or she will check your all the biochemical parameters. So, and lifestyle information, he or she will ask that whether you are having a sedentary lifestyle or whether you are very active, you are a sports person, or you are this, you are that, everything will be asked by the doctor. So after that, he or she will use the algorithms provided by different AI ML softwares, and that algorithm will let you know that whether there is a chances of increased risk of a particular disease, like let's say type 2 diabetes mellitus, let's say fatty acid liver disease. And in this case, it is predicting that whether this patient is capable of suffering from atherosclerosis or not. Atherosclerosis means the choking of the arteries, the blockage of the arteries with the cholesterol and lipid based thing. So all those things will be provided. Identify critical biomarkers and microbial profiles for predicting atherosclerosis risk. So algorithm will be run, and after the successful completion of the algorithm, it will provide the biomarker that it has this in whenever you go for a diagnostic test, let's suppose sonography. So in sonography, let's say ultrasound, what the reports says. So try to have a look on all those things, because it will provide you a lot of information. So it generally give the comments, and after that a final interpretations are mentioned below the report. So it is like the patient is having some cyst, abdominal cyst, or this and that. So this cyst can be turned into something very dangerous or not. It is written down, or else if it is required that some more confirmatory tests are required or not, so it is also recommended that advice this test is recommended for confirmation. So accordingly, this algorithm will provide you that the patient is having this microorganism which can cause the increased risk of that disease. It will predict you. So that risk prediction is done by the algorithm. And after that, you can if this has provided that the bacteria A can turn you into a heart CVD, cardiovascular disease. So what you can do, you can directly target that. Target identification is done, then you are good to go. Because doctor is always in a need of identifying the source of the disease, the root cause of the disease. Because whenever the disease is no root cause of the disease is known, then you can be you are able to cure it. So it's heavily raining here, and it's a very thunderstorm, and I'm so scared of, so background noise is disturbing me. So you can say if the risk is provided by the algorithm, you can directly target that. That can be easily targeted, and then you can have personalized interventions, like you can have gut boosting diet, you can have prebiotic, you can have probiotic, and precision therapy. So I hope the things are clear. Like if you are providing all your data to the algorithm, then algorithm will identify that yes, this has this biomarker and this can increase the risk of this particular disease. Now you are having a target, you have to target that antagonist, you need to do some uh, personalized medicine to kick out that particular biomarker. So these things are going on in the AIML in microbiome research. So this is the summary of whatever we are having in. You can go for live biotherapeutics, you can go for personalized therapeutics, and for data mining we use AIML, for sequencing data mining we use, and for predicting microbial biomarker discovery. So power cut. It's very difficult nowadays in Kolkata, particularly in Kalyani, because it's raining since last 2 to 3 not since Sunday it's raining heavily, and I am so fed up of everything. Like everywhere it is very uncomfortable to go. Now we have come to the last topic of today's session, you can say, or fifth module, you can say. In this we are having the most important thing. If you are going into any kind of medical based research or medical research, then in the interview also people will ask you the ethical and the regulatory compliance associated with any kind of medical research or any kind of clinical trial. So people will directly ask you, do you know about the ethical and the regulatory things associated with this and that research? So every time, whenever we talk about the microorganism, whenever we are talking about therapeutics, so whenever I write micro, I usually write mo. So I don't know you guys are able to get it or not. Mo means microorganism. I only write mo. So I just now realized that whether you are able to understand or not. So mo means microorganism. So microorganism based, if you are doing any work or if you are doing any therapeutic based work or any clinical trial, then you cannot forget this word ethical issues, ethical concern. Uh, so basically, in ethical issues, each and everything should be in a written form. You cannot say just I have informed the patient verbally, orally. No, you have to take his or her written consent. And in short, everything needs to be revealed. Uh, you can say up to 100% original thing. You should be doing the original. You can say you need to reveal the consequences of, if you are analyzing any drug or any therapeutics, you want to go for the clinical trial into human subjects. So for that, what may be the consequences of that particular drug on the patient? You cannot hide that. If you are hiding that, uh, assuming that if you will let the patients know that these are the consequences, you can get this, you can get that, so the patient will withdraw its consent. So you are doing an unethical thing. So in research, whenever a people is found that he or she is associated with some unethical thing, so they are fired on the spot. They are fired, because all these things are very very important, because all these things are directly related to a person's survival. If you are hiding anything from that particular subject, uh, who is a very important part of your clinical trial, then you are taking his or her life very lightly. This is not the thing to do so in clinical trial. So the first and the foremost part is informed consent. A written informed consent is there, and in that, patient must fully understand what the source of microbiome. As we are studying the ethical and regulatory aspect in the context of microbiome therapy, so we always take the informed consent of the patient. Like these are the sources of microbiome. Example, from which donor you have taken the feces. You also have to reveal this part also, that you have derived the feces from this patient, and you also have to provide the all the uh, uh, biochemical results of that donor to the patient also, because he or she has the right to view the reports of whatever donor feces he or she is transplanting. So everything you need to reveal it to the patient. So you need to mention the source of the microbiome, like this will be the donor of your FMT. And the next thing is risks and benefits. What are the risks associated with the FMT, like fecal microbiota transplantation? Like you can have this anxiety, depression, or you can have some gut related issue like digestion related issues. And what are the probable benefits? So you have to provide all the pros and cons. Now, possibility of long-term side effect, you will also let them know. Let them know means you will explain them. And also nowadays, not nowadays, it is since so long that whenever there is a patient, you need to explain them in their local language also, local regional language. Uh, if you are explaining to them not just in English, some people will understand and some will not. So if you are explaining them in their local language or their regional language, so people will be comfortable to understand it in a very clear way, and you can communicate easily. The more you will communicate with the patient, the more they will be able to reveal their symptoms also to you, and they will get your point very clearly. So all it depends on doctor will let you know all the things in a very uh, good way, in a very comfortable communication he or she will let you know. So you will let them know all these things. And the special concern are taken for whom? The people, pediatrics, like for the children's, for the immunocompromised population, like if the people are older, if they are immunocompromised, if their immunity is very low, if the people are suffering from any autoimmune disease. So that are some special concerns which the doctor will take care of. Now B, which is again important part, privacy and data protection. So this is the you can say the most important part, or you can say the heart of the ethical, where the ethical concerns arise. Like you cannot leak the data of a patient. You can show the data, suppose this is the data like xyz person, and these are the profile of its, let's say, liver profile, kidney profile, and all those things. So you cannot reveal this in a public database mentioning his or her name. No, you cannot do that, because this is the you can say this is my private report. How can I reveal my private report to anybody? So this might be a concern for people who don't want their data to be shared, and many of the majority of the person never share their data. So if whenever you are using, so in research what we do, we compile all those clinical trial data. So if you are aware of that or not, in that no patient name, gender is mentioned, but no patient name or anything related to identification is there. It is directly like 30 human subjects, 40 human subjects, and age will be also mentioned, age, and if they are having any kind of metabolic disease or not, like diseased one, normal one. This type of things are there in the uh, paper. So you cannot reveal their personal disease profile data to anybody. So microbiome data can indirectly reveal genetic, lifestyle, or disease information. So you cannot leak that. Risk of re-identification from metagenomic sequencing data. So now donor screening. Screening means how you can fix if you are having five donors are there that are willingly giving their feces for the fecal microbiota transplantation. So how you can choose out of the 5 person to take the feces from out of the five, from 1, 2, 3, 4, 5, which one? So there are different ethical dilemma associated with that also, to whether I can take from one for first person, second person, third person, fourth person, and likewise. So how much screening is required to prove that three is more appropriate to give the feces? So there are so many ethical dilemma associated with it. Now risk of transmitting unknown pathogens. So whenever we are going for any kind of surgical process or any kind of uh, medical related thing, so there are always chances of getting some unknown pathogens. You cannot say, because now uh, you suppose pseudomonas also, you know that this readily grows on the catheters, medical implants, and all those things. So this pseudomonas has a potential to form biofilm on any of the medical devices like implants, catheters, wounds, anything. So if you are going into an hospital where there is an already a pseudomonas based infection, so there are higher chances that you might be infected with that pseudomonas or any of the pathogen. So all these things are having these secondary complications, like you may get infected. So people take withdraw their consent that no, I will be not giving the my any thing to for the transplantation process. So all these things are very risky also, you can say. The donor's medical and lifestyle history must be thoroughly assessed. So whenever you are taking anything from a person, obviously you have to know about its medical history. If he or she is suffering from any kind of disease in the past, then it may have some impact on its gene, on its microorganism diversity and everything. So lifestyle history also, whether he or she is eating only junk, so its microbiome profile will be something different, whether he or she is in on antibiotic or not. So every little information is required to remove the dilemma that whether I have to choose this one or that one. So that dilemma has to be removed. Now, manipulating microbiota. So concerns over playing with the natural microbiome. Now, always in biotechnology, you will be in a very, you can say, a lot of ethical concerns arises whenever you are manipulating a normal microbiota. Whenever you are manipulating any bacteria, because in our research we consider each and every organism as important as a life of a human. We cannot say that bacteria hi to hai, no. These are all unethical things. So whenever we are modifying a bacterial genome, a very you can say wide range of concerns arises, that whether this bacteria will be mutated, it can be released into the environment, so it may lead to bio war, bio weapon, or likewise things. So whenever we are doing any kind of work related to bacterial genetic engineering, so before releasing it into the market, or you can say if you are not synthesizing or manufacturing any product, you are doing your own PhD research or own postdoc research. So then also people will always say that where you dispose of your bacteria, because whenever it is left into the environment, it may get mutated, it may get very dangerous and pathogenic for each and every living organism. So for that, so many concerns arise that whether the manipulation is fruitful or not, because it may lead to the increased pathogenicity of that particular bacteria. Because whenever you are converting any wild type to mutated type, so you cannot say that this will be always in a good mood. No, it can lead to some increased pathogenicity. So manipulating a microbiota is always a concern, because you are manipulating the genome of a natural microorganism. So that way also it is an ethical concern that you are manipulating the genome of a you can say a living organism. So always it will be a concern. Now, as I told you, possibility of unintended ecological disruption, new pathogens or resistance gene. So as I told you, people will always ask you that where you dispose of your bacteria or whatever microorganism you are handling after your completion of the research process or your research experiment, what you do with your bacteria. So this is a very obvious question. So you cannot directly flush into your sink, because that is connected with the normal water bodies. So if you are disposing of your microorganism directly into the sink of your laboratory, then you are contaminating the water bodies associated with that particular uh, outlet. So you can uh, you can, what you can do, we need to very carefully, we need to discard that particular bacteria. So always in laboratories we have biohazard bags, red color bag and black color bag. We have different color bag to uh, uh, safely dispose of all our pathogenic things in our lab. So these are also a ethical concern and a regulation that you must dispose of in a very safe manner, that it should not contaminate any kind of water bodies, or it may not contaminate the soil also, because soil-borne pathogens are also there. And equity and access. So what is this equity and access? Will advanced microbiome therapies be accessible to marginalized population? Avoid creating a microbiome divide similar to digital divide. So equity and access means whether it will be accessible for all of the people, or whether it will be divided for this much population. So all these things are related to whenever you are working with a gut microbiome based therapy. Now, what are the regulatory challenges? So all these are ethical challenges. Now, what are the regulatory challenges associated with the fecal microbiota transplantation, you can say, or whole gut microbiome therapy, you can say? So lack of global consensus, no unified regulatory framework globally for FMT and LBP. LBP is live biotherapeutic products. So you can say there is if you do research. So what we do trial and error. For some people, if they are associated with any kind of research which is done since so long, so already all the protocols will be standardized, every concentration and every experiment will be standardized. So they only have to blindly follow the protocol and they will be good to go. But for many of us, including me also, we don't have any standardized protocol, so that always take very time taking, because we have to standardize each and everything, like concentration of each and every reagent, the timing, uh, all the molecular based thing, we have to just go for the trial and error. So in uh, microbiome based therapy also, we do not have any standardized protocol that you have to follow this one, stick to this one, you will get the result. No. So this creates a little bit of mess, that we don't have a unified regulatory framework, that we have to follow this much of and this much, and that way we have to go. So for FMT and LBP, we do not have any unified regulatory framework. So for fecal microbiota transplantation regulation, we have US FDA and EMA and ICMR in India. So you can say US FDA classified as a biological drug under IND, that is investigational new drug regulation. So temporarily allowed for, as we have studied, the Clostridium difficile disease. So fecal microbiota transplantation is for CDI. Now, suppose in India we are having ICMR, so Indian Council for Medical Research. So we are having guidelines provided by ICMR, and we have to stick to the whatever guideline provided by the ICMR. So if you are looking for some job opportunity, you can say, so ICMR always readily recruit so many different level people, like graduate, postgraduate, PhDs, postdocs. So you can check it out, the official website of ICMR. So they always provide a very good opportunity. You can always have a look, because they readily recruits. Like it is not so that vacancy will come in only this month, and not after 6 months they will hire. So you can check that out. So in India also, we are having guidelines by the ICMR related to stem cell and biotherapy rules. So we have to stick to whatever be the regulatory framework provided by the our own country medical unit. Now, product standardization. So this is also again a very important thing. Like we have to go for the standardization we have to do a lot of experiment before standardization. Some experiment will work, some experiment will not work. So you have to determine that this I was talking about. The defining dose means you have to decide that this much of bacteria you can give. So in bacteria, we talk about, we talk in terms of CFU, colony forming unit, like 1 into 10 to the power 6 CFU per ml. 1.8 into 10 to the power 4. So likewise, we talk in terms of CFU per ml, that how many colony forming units can be there in per ml of bacterial culture. Quality control, is it pure or impure? Is it contaminated or what? Shelf life, is it active for 6 months, 1 year, 2 year? Everything you have to mention on your product. So always it takes time for the standardization procedure. Now safety and efficacy. So long term clinical trials you need to do, because whenever you are declaring that this drug is not having side effects, so you need to have long term clinical trial like 6 years, 3 to 5 years. So likewise, then only you can say that this is safe or this is efficient. And also through that you will be able to predict if the host will develop some resistance to that particular antibiotic or not. So it is all related to the microbiome complexity, what kind of microorganism we are having in our gut. Now the most important one, intellectual property right. uh, who owns a strain isolated from a human microbiome? Issues in patenting natural microbes. So see, we cannot patent, people are always confused, we cannot patent the microorganisms. No, why? Because this is a natural thing. Patent is only applicable for the things which are not obvious, not so obvious. It means if you are adding water and sugar, and you are saying that this is a sweet solution, and you want to do the patent for this is an example for explaining the concept. So if you are adding water and sugar, and then you are saying that I have prepared the sweet solution by adding water and sugar, so it will not be patented, because this is an obvious thing. If you are adding sugar, then it will be sweet. If you are adding salt, then it will turn salty water, saline water. So if the result is obvious, it means it is decided that this result will come. So you cannot patent that. Always patent should be non-obvious, that non-obvious things are patented, like you got some any different result, and apart from that you cannot go for any kind of natural thing to be patented. No microorganisms you can patent, no natural plant you can patent. Yes, you can patent what? If you have genetically altered this microorganism, you can patent that. You can patent the process of any thing which you are doing. Like if you are doing the manipulation by your process patent can be done, and then you get royalty if you sell your process to any of the industry. People do that. So you cannot patent the natural microbes, you cannot patent anything that is naturally existing on this earth, because you have not contributed anything to that, that is existing in your environment naturally. So if the things in which you don't have any kind of contribution, you cannot patent it. All these things are intellectual property right. So in IPR also there are so many vacancies. You can try, if you want to be a patent agent, you can go for the patent examiner, what that exam name I forgot. So there are some patent officer exams conducted by PSC. Central level examinations are there, and state level are also there, so you can apply, because these positions are also have a very fair pay, so you can go for that. These jobs are very you can say a stabilized job. So a patent can be done if you are having your own contribution to that, and you are getting non-obvious result. So key guideline and organization, who provides the guideline that you have to stick to this procedure, you have to follow this one, you have to provide your result in this way, that way. So it is provided by US FDA, Europe EMA, ICMR India, and of course we cannot forget WHO, World Health Organization. So everybody provides their own regulatory guidelines. We have to stick to there. Without having the clearance certificate from the each and every organization where we are residing. You cannot go for the either patent, or even you cannot conduct your clinical trial without people always you have heard that are clearance nahi mil raha hai. So all these things are because you are not sticking onto the whatever guidelines provided by your organization. So whenever you are 100% satisfying the criteria mentioned by the WHO, ICMR, or wherever you are residing, so then only you will be getting a clearance certificate, and then you can go for your clinical trial. Without any ethical clearance, if you are conducting anything, uh, you will be punished. So future ethical concern will be designer microbiome. So people will always genetically manipulate the microorganism, because obviously wild type have some sort of restriction. So people will do what? Alteration in it. CRISPR is a very you can say required and need of the hour nowadays. And biobanking of fecal sample. Biobanking means people will, like blood bank, likewise biobanking of fecal samples. You people will collect the fecal sample for future use. They will freeze it in -196 liquid nitrogen, and then you can uh, you can provide its ownership, and you can use it in future. So all these things will be happening. Now these are all the summary, like what is the ethical and regulatory aspect in microbiome therapy. So there are so many things. We have to take informed consent, we have to remove the dilemma that we have to choose which donor, and if we are manipulating any microbiota, so our aim should be without any secondary concern that may arise if it is left exposed into the environment. So we have to take care of the proper disposing of. So all these are safety guidelines. Safety guidelines means whenever you go for a PhD research, your research institute will always ask you to go for the safety guideline workshop, and then you have to pass a test. You have to do basically a certification course that how to do work in a laboratory. So every research scholar go through this safety guideline in his PhD, and it's sometimes postdoc also. So in this safety guidelines, people will taught you how to do any research experiment in lab. Like what are the safety measures you need to take? You have to wear the PPE kit. You right from the PPE kit to safely disposing of whatever you are using in your lab. And if you are having some fire related work in your experiment, so what to do. And if you are having some radioactive substances, so how to handle that also. If you are having some carcinogenic substances in your research, how to handle that. So everything is provided as a safety guideline. It is obvious like 2 to 3 days training. So people will let you know how to handle that, how to protect yourself while conducting the experiment. So all these things are under the safety guidelines workshop. So all these things are there. So some questions are also there, but I think there is no time, and already it is heavily, so I'm not like to extend more. So we will solve this question tomorrow also. Let me stop the recording. We will solve some more questions tomorrow.